Welcome back to the lecture series of overview and integration of cellular metabolism. We are at week 8, lecture 40. We are going to discuss tryptophan metabolism in this class. So, the concepts here covered are metabolic fate of tryptophan, different metabolic roles imparted by tryptophan. Then there are different specialist products which are synthesized from tryptophan, their roles, and the associated clinical conditions with defective tryptophan metabolism. Tryptophan is one aromatic amino acid, essential amino acid because it cannot be synthesized in our body. Then it is a glucoketogenic amino acid because alanine is produced from it, and ketogenic because acetoacetate is produced from it. Then there are different important biomolecules which are formed from tryptophan, like serotonin, which is a neurotransmitter, niacin is a vitamin, and melatonin is a hormone. Remember, this is not melanin. Melanin is a pigment, whereas melatonin is a hormone. Please concentrate on this. Okay. Now, we are moving on to catabolism of tryptophan. Now, remember, catabolism of tryptophan, the whole pathway might not be that much important, but the product kynurenine is important. Here you can see kynurenine. Now, kynurenine, more than 95% of tryptophan are actually metabolized, rather catabolized to form kynurenine, and in this regard the enzyme pyrrolase is important. Now, pyrrolase can be indolamine 2,3-dioxygenase, which is present in immune system and brain, or can be the variety of tryptophan 2,3-dioxygenase, which is present in liver. Both of them, based on the tissue specificity, present in different tissue, differential expression in different tissues. This is the most important enzyme for breakdown of tryptophan. Now, tryptophan, it forms N-formyl kynurenine with pyrrolase, then forms formylase, where you can see formate is released and kynurenine is formed. Then kynurenine undergoes steps of reaction, series of reaction forming hydroxykynurenine via the enzyme hydroxylase. Then it undergoes kynureninase, where 3-hydroxyanthranilate is formed. Now, this step of reaction is basically a where the aromatic ring is actually opened up. Here you can see the aromatic ring is opened up while forming 3-hydroxyanthranilate, and what is released is alanine. That is the reason we say this is one glucogenic amino acids. And this kynureninase enzyme is vitamin B6 dependent enzyme. Now, here you can see 3-hydroxyanthranilate via the enzyme oxidase undergo opening of the ring, forming acrylyl amino fumarate. So, remember, kynurenine undergoes ring opening via the enzyme kynureninase and oxidase, where molecular oxygen is also required. Now, it finally, this acrylyl amino fumarate finally, following different series of reactions, form acetoacetate, which is the ketogenic form of ketogenic product of tryptophan. So, regarding the kynurenine, which is one very important metabolite of tryptophan, it has few correlations with different diseases, like cognitive deficits in schizophrenia. It is associated with the disorder in kynurenine degrading enzyme. Similarly, the levels of kynurenine is found to be decreased in bipolar disorder, or the production of kynurenine is increased in case of Alzheimer's disease or cardiovascular disease. Also kynurenine is associated with different movement disorders or tics. So, these are the association of kynurenine with different diseases. The metabolic role of tryptophan, the important products are NAD, nicotinamide adenine dinucleotide, from which NADP, nicotinamide adenine dinucleotide phosphate is also formed. So, along with NAD there is NADP also. Then serotonin as well as melatonin, these are the important products formed from tryptophan. Now, to remember, indican is one such metabolite or biomolecule which is formed from tryptophan during its catabolism and is excreted in urine. So, when there is excess tryptophan catabolism, indican level in urine will be high. Next, I am coming to the synthesis of NAD and NADP. So, basically from tryptophan we have seen how kynurenine is formed, how hydroxykynurenine is formed, and finally 3-hydroxyantranilic acid is formed. Now, from 3-hydroxyantranilic acid there are multiple steps following which quinolate is formed. Now, this quinolate undergoes phosphoribosylation. So, this phosphoribosyle group is actually coming from PRPP, phosphoribosyl pyrophosphate. PRPP is donating the phosphoribosyl group to quinolate. So, here you can see formation of nicotinate mononucleotide due to transfer of phosphoribosyl group by the enzyme phosphoribosyl transferase. Now, AMP is donated from ATP. So, here you can see there is one adenosyl group, there is one phosphoribosyl group as well as there is another phosphoribosyl adenine group present, forming des-amidonicotinamide dinucleotide. These undergo transamination. So, glutamine donates the amine group to it. So, here we are getting one ribosyl phosphate, adenylated ribosyl phosphate, along with that other ribosyl phosphate. So, that is why it is known as nicotinamide dinucleotide. So, NAD is formed here. Along with that, NADP can be formed from NAD via phosphorylation. So, here you can see these two phosphate groups were there already. Now, another phosphate group phosphorylation occurs in NAD to form NADP, nicotinamide adenine dinucleotide phosphate. So, this is how NAD and NADP are formed from tryptophan. Then we are moving on to serotonin synthesis. Serotonin is a neurotransmitter and that is formed from tryptophan via hydroxylation and decarboxylation. Now, tryptophan on hydroxylation form hydroxytryptophan, that is 5-hydroxytryptophan. The enzyme is tryptophan hydroxylase, and like all the hydroxylases there is tetrahydrobiopterin, which forms hydroxy tetrahydrobiopterin. So, we are getting 5-hydroxytryptophan. 5-hydroxytryptophan undergoes decarboxylation with the enzyme decarboxylase. Like all the decarboxylase, the cofactor is PLP or pyridoxal phosphate. So, on decarboxylation there is formation of 5-hydroxytryptamine, and that 5-hydroxytryptamine is basically known as serotonin. So, this is how serotonin is formed from tryptophan by one hydroxylation, then one decarboxylation reaction. Now, the formed serotonin is degraded via the enzyme monoamine oxidase, and the product is 5-hydroxy-indole acetic acid, which is excreted in urine. So, this is how serotonin is formed and broken down. Now, we are moving on to melatonin synthesis. Once again, to remind, melatonin is a hormone, whereas melanin is a pigment. We are talking about melatonin, the hormone. So, melatonin hormone is basically related to our circadian rhythm. Sleep awake, sleep cycle pattern is regulated by the secretion of melatonin. Now, melatonin is synthesized in pinealocytes, and the major part of melatonin synthesis occurs in the dark phase of the day. So, while it is exposed to the light, there is basically proteasomal degradation which causes the melatonin, which halt the melatonin synthesis. Now, for melatonin synthesis, the important enzyme is serotonin N-acetyl transferase. So, from serotonin, which is 5-hydroxytryptamine, the enzyme serotonin N-acetyl transferase forms acetyl serotonin, which undergoes methylation to form melatonin. So, this is the formation of melatonin hormone. Now, remember, this is melatonin synthesis. Melatonin is degraded or catabolized by forming 6-hydroxymelatonin, which is conjugated and rather solubilized by sulfation, that is adding sulfate group. It is solubilized and then it is excreted in urine. So, this is how melatonin is synthesized and degraded or catabolized. So, here we are to discuss the defective tryptophan metabolism related disorders. These are the disorders I am going to discuss: Hartnupp's disease, pellagra and carcinoid syndrome. Now, remember, pellagra is basically dietary niacin deficiency, but because tryptophan is important precursor for niacin synthesis, that is why tryptophan deficiency is the tryptophan deficiency is manifested as pellagra like manifestations. Then Hartnupp's disease. Hartnupp's disease, we already have mentioned this in the absorption and digestion of protein metabolism, the very first class of protein metabolism. Let us discuss a bit in details. So, Hartnupp's disease is basically one autosomal recessive variety of disease, is an inborn error or inherited disease. This occurs due to mutation in the gene SLC6A19, which encodes for the protein B081, which is also known as BOAT protein. Now, BOAT1 protein is basically a sodium dependent co-transporter, which is present in lumens of gut, small intestine, also in kidney. So, where those proteins are located specifically, those are in the apical surface of small intestine as well as the lining of renal tubular cells. So, those are the transporters for whom? Neutral amino acids. So, what will happen? The transporter for neutral amino acid is not working in small intestine. So, absorption of those neutral amino acid will not occur. So, what will happen to those which are present in the diet? They will be excreted in stool, feces. Then also neutral amino acids which are actually filtered through glomerulus, they cannot be reabsorbed in kidney, because once again the transporter is absent in renal tubular cells, the site for reabsorption. So, what will happen once again? Urinary excretion of those neutral amino acids will be there. Now, amongst the neutral amino acid there is tryptophan. Now, remember once again tryptophan is the precursor for niacin. That is why niacin deficiency will be there and will be manifested as the symptoms of pellagra, like skin eruptions as well as neurological symptoms, dermatitis, ataxia. These are the manifestation which are due to niacin deficiency basically. Then because there is urinary excretion of the neutral amino acid, a generalized amino aciduria will be present. So, there is excretion of neutral amino acid in urine. Also the neutral amino acid in gut, they will be excreted through colons. Now, there is colonic bacteria which will act upon those compounds, will convert those in indolic compounds, which will be conjugated in liver and will be excreted in urine as indican. Remember the metabolite indican that is excreted in urine in during tryptophan catabolism. So, indican will be excreted in urine. So, how this will be diagnosed? The commonest diagnosis is a generalized amino aciduria, where the amino acids, neutral amino acids can be detected via chromatography in urine. Now, proline has a different proline, though it is a neutral amino acid, it has a different amino acid transporter. So, basically in the generalized neutral amino aciduria, proline will not be there. Who will be there? Valine, serine, phenylalanine, histidine, glutamine, leucine, asparagine, all these amino acids along with tryptophan will be detected in urine. Also indicans will be also present in urine. So, what will be the basic line of treatment? Because there is neutral amino acid deficiency, that should be supplemented as much as possible in diet via providing high protein diet, as well as because there is manifestation of niacin deficiency, because tryptophan is not there. So, niacin should be supplemented in diet. So, these are the line of treatment when there is heart nerves disease. Now, we are moving on to carcinoid syndrome. Now, carcinoid syndrome is a type of paraneoplastic syndrome. Now, remember what is paraneoplastic syndrome? Paraneoplastic syndrome is the manifestation of malignancies, but majority of the not majority, but mostly the manifestation of malignancies are coming from the malignant cells, mass actions or different other functions. But paraneoplastic syndrome, those malignant cells secrete some biomolecules which exerts their chemical function or endocrine function over body. So, those functions, those biomolecules related functions which are generating from malignancy, they are known as paraneoplastic syndrome. So, carcinoid syndrome is one such paraneoplastic syndrome, and the malignancy here is neuroendocrine tumor, mostly argentophenoma, tumors of argentophin cells, and that malignancy is mostly located in gut. Common regions are appendix, stomach, rectum, colon, pancreas, as well as apart from gut, those are also can be located, those tumors occurs in lungs as well. Now, what are the manifestations? Because excess vasoactive mediators are released, apart from serotonin, histamine, collicrans, these are released, there are excess gut secretion also excess secretion in bronchioles, in the lung variety. So, what happen? The manifestations are diarrhea, hot flashes, asthma, wheezing, pain abdomen. So, these are the manifestation due to release of vasoactive mediators like serotonin, histamine, colchicine, like that. Apart from that, once again there is pellagra like symptoms. Why? Normally tryptophan, around 1% of the tryptophan are actually converted to serotonin. But here in carcinoid syndrome, around 50 to 60% tryptophan are actually utilized to form serotonin. So, basically the production of niacin will be decreased, causing these pellagra like symptoms. The diagnostic mode is basically imaging, via which the tumors to be detected, and also in urine 5-hydroxyindole acetic acid can be detected. So, these are the different biologically important substances we have discussed regarding tryptophan metabolism, like alanine and acetyl coenzyme, acetoacetyl coenzyme. These are the catabolism related products. Also formyl group as released as formate, which can be utilized as a 1 carbon unit in different metabolism. Then niacin and nicotinamide adenine dinucleotides as well as NADP are also important. Then serotonin, melatonin, those are also formed, and as excretory product of hydroxyindole acetic acid and indicans. These are the important products from tryptophan. So, the key points are tryptophan is a non-essential amino acid. So, basically synthesis in body is not there, whereas catabolism is there. The main product is kynurenine, which has relation to different diseases, and also other metabolites like serotonin, melatonin, niacin, NAD or NADP. These are the different derivatives from tryptophan. These are my references, and see you all in the next class. Thank you.